We're very excited for this talk today. Today, the title of this talk is Advances in Islet SEM Scanning Below the Membrane, presented by Drs. Jing Hughes and Ben Giepmans. So just a short bio for each of them. Dr. Hughes is a physician scientist and an islet biologist at WashU in St. Louis. Her work focuses on the islet cell surface sensory organelle primary cilium. Primary cilia are antenna-like organelles, most people probably understand this, that coordinate cell signaling, where mutations in ciliary genes are linked to human metabolic diseases, including diabetes. Her team uses genetic models and advanced imaging to study primary cilia structures and function in rodent and human islets. For this talk, she's going to present recent work by her lab in developing a new method of scanning electron microscopy, or SEM, to study the submembrane structure of islet primary cilia, and findings that have shed new light on cilia functionality as a signaling device in glucose regulation. Dr. Hughes is an NIH-funded investigator and a member of HERN, as well as participant in 2-1D exchange and rare and atypical diseases network radiant clinical trials. For Dr. Giepmans, Ben Giepmans is intrigued by how biomolecules act together to control cell fate in health and disease. He's a cell biologist and molecular biologist microscopist. His PhD research at the Netherlands Cancer Center and related work in the Scripps Institute have led to better molecular understanding of gap junctions. In his second postdoc, he implemented several new advanced imaging techniques and probes to study protein dynamics in live cells and protein localization at high resolution. These studies have given unexpected new insights in Golgi apparatus reformation during mitosis. Type 1 diabetes and innovative microscopy are the spear points of the Giepman Lab Research Group at UMCG Groningen, where he also heads the Advanced Microscopy and Imaging Center, UMIC. The team develops and or implements new imaging techniques and with and without probes for a large-scale and multimodal microscopy. Particular focus is on the islets of Langerhans to help understand triggers and potential new therapies for type 1 diabetes. Welcome to both of you, and thank you so much for joining us. I'm very excited to hear more, and I think we're going to start with Dr. Hughes.
Okay, thank you, Monica. That was great. Happy to be here. Very honored to be paired with Dr. Giepmans, and whose work I've always admired, actually, as a clinical endocrinologist. He's done phenomenal work on T1D research and the human islet morphology. So yeah, as Monica said, I'm an islet biologist here at WashU. My team actually focuses a lot more on cellular function signaling, but this is kind of a side project that just for indulging, playing with new tools here on campus. We wanted to see what how far we can push the resolution for imaging this particular organelle that we're studying functionally, which is called the primary cilium. And just a very quick introduction for those who don't really think about cilia 24/7 like we do. It's a very important sensory organelle, and pretty much every cell on the body has one, with the exception of a few very specialized cell types. We have primary cilia in every cell, which carries a key function in sensory and signaling. It's really called the cellular antenna, and for good reason, because it is responsible for picking up a variety of cues, including mechanical, chemical, and in a closed environment like the islets of Langerhans, the cilium is really a communication device between islet cell types and also different cells within the same cell type. And there's a lot of unknown functions that primary cilia play in islets, which we're trying to uncover, but also at the same time, we really need to know what they look like and what are their morphological features in order to even start to have hypotheses about what they might be doing. So my team has been focusing for the last few years, developing new microscopy methods to really push the envelope and seeing how well we can visualize these organelles. Now, there are limitations with both light and electron microscopy imaging of cilia. With light microscopy, you really need to know what you're trying to label in order to see it, right? It has to be antibody-driven, protein-driven identification. Transmission EM is actually the classic method for studying cilia, and I've shown a few slides here. Some of them are our unpublished TEM work. And that is good for an unbiased survey of cilia morphology. But the issue with TEM is that it's actually exceedingly difficult to find these things in a TEM slice of, say, a human eyelid tissue slice or tissue block. And the problem with studying cilia is that depending on who is doing it and where it has been sampled, I said like where in the cell, the cross-section actually will show a different arrangement of the ciliary components and these microtubules that build the stalk of the cilium. And so the conventional model of cilia structure is actually still somewhat in dispute. And that's a question we set out to answer. And what do actually human eyelid cilia look like? Do they belong to this conventional classification of primary cilia versus motile cilium or something in between, like a hybrid? Like I said, hunting for these things by TEM is difficult. And so we have been looking for a few years for an alternate approach to look at these things both unbiasedly, but a little bit easier, hopefully, for identification. Like I alluded to, the ciliary structure differs depending on where along the cilium axoneme you look. And so this is an issue that we sort of encountered trying to do FibSim, and also other groups using FibSim have reported, depends on where you slice, the cross-sectional arrangements are different. So we really wanted to get kind of a top-down view and also just to scan the full length using either tomography or, as I will say today, a new approach using scanning EM, which allows visualization of the entire structure on the surface of the eyelid. And so the work I'll be presenting briefly today is newly published, and I put the PMID number on the bottom of the screen, so those of you interested can look it up. Just came out a couple of weeks ago. And we wanted to remove the sampling bias of TEM by looking at the full length of the cilium, and then to start correlating things with what we're seeing by light microscopy. So a brief overview of the process is actually pretty labor intensive, but I've simplified the workflow here. The imaging is done actually on a FibSim apparatus that we have here at WashU. This is an stock image, not our machine, but it looks very similar. It basically involves taking whole donor eyelids from healthy human donors, attaching them to a plate and then fixing, and also at the same time demembranating. So we actually peel away the surface of the cell in order to reveal the cytoskeletal components, both on the cortical surface, but also underneath the ciliary membrane. I have a few slides I can show you what it looks like before and after demembranation, and it's really this key step that is new in our workflow that allows visualization of the ciliary components. So here's an intact eyelid using our method of demembranating and then imaging on a fixed glass cover slip. And you can already see that there are different cell types that can be highlighted based on their surface morphology alone, which is beautiful to look at. And so drawing from the literature, based on what we know, different cells have different types of organelles distributed on their surface, we can start to identify, alpha versus beta cells. Beta cells have these beautiful uniform microvilli that are covering their surface. It increases the surface area for things like glucose sensing. Alpha cells are relatively, have less abundant in terms of microvilli. Delta cells have longer filopodia, but then all of these cells have a primary cilium. Not every cilium gets preserved after the workflow, so you can see some of these cells don't have a visible cilium, but those that do, we can then zoom in and take a very close look. Here's what it looks like before and after demembranating. On the left side, the native eyelid surface, you can see some features. Certainly the cilia is visible, but you really don't gain an appreciation of the microtubule structure until you've lifted the membrane off. And that's the picture on the right. So with this technology, we can start to look at morphology, start to count microtubules, start looking at the length, the width, and then importantly, there are secondary structures that these microtubules undertake, which would not otherwise be visible without demembranating. So on the right side, I've shown you a couple of examples of these microtubule bundles that arrange themselves into a chiral helix, and we can start to quantify whether these are left or right-handed turns. in the human islet, it typically is a left-handed helix, and we don't quite know what that means in terms of the biomechanics. But there must be important functional implications that we're going to be looking for. Axonemal structural details are revealed by this technique as well, and we were really excited because things that have only been described anecdotally on TEM has now been revealed, and you can just get such a better view and better appreciation for what things look like. For example, there are actin attachments at the base of the cilium. This has been well described by a number of groups, but only using TEM, and now we can actually see how they're arranged and attached to the ciliary stalk in 3D. The microtubules themselves are beautiful, and you can see their chirality starting at the base. The ciliary pocket, where the ciliary axoneme grows out of the cell, is also captured pretty well, and you can sort of appreciate there are appendages that sort of anchor the axoneme below the cell surface. Things like doublet to singlet transition of the microtubule filaments are captured as well, and this is important because it has implications for ciliary flexibility, structural rigidity, and what we also recently described, which is the ability for these things to move in the 3D space. And we think the movement may be enabled by these structural changes along the length of the cilia. The ciliary tip is a very special region, which we have a special interest on. It's a signaling hot zone, and so we wanted to just take a look at the morphology. It's beautiful, and it has a very striking different texture than the rest of the cilium. We don't quite know what is the protein composition, for example, at the tip, and that is something we're going to be working on with immuno-EM approaches to identify. But also, interestingly, like I alluded to, these things are motile, and the tip region is particularly dynamic. And you can see in this live video that where we've labeled the entire cilium with a GFP molecule, the tip is quite motile, particularly relative to the base. And we can actually start to trace the trajectory of these things in space and analyze how their motion is affected by things like ambient glucose, other nutrients, etc. So that's a quick overview of what we've been doing in terms of islet cilia EM, particularly scanning EM. And we do have plans to go further with this protocol to look at differences between healthy versus diseased donors, particularly in diabetes. What happens when, say, chronic hyperglycemia conditions, which have been known to kind of impair ciliary function, what does that do to the morphology? Or maybe vice versa, could be a chicken or the egg. And then also differences between species. So far, we've done both human and mouse islets, and we're looking into the differences between cross species on ciliary composition and the microtubules. And then, like I alluded to, individual protein identification using immuno-EM is something that we are actively working on. And we have some exciting pilot results. Hopefully we can share next time on what kind of signaling proteins may be residing on the islet primary cilia to allow its function as a signaling organelle. So that's it. And the work that I talked about today is done primarily by these 3 team members. Sonia was part of the Wookiee Imaging Center, which is pictured below in this image. Alex was a postdoc and the lead author on this work. And Bella is my student from the MSTP program here at WashU. Thank you. And I'll take any questions at the end. Thank you.
That's fantastic. It's really beautiful. And I guess I will ask at the end if there's any postdoc, projects available for both of you, so we can elaborate then. And now let's sort of shift gears for a minute and go to Dr. Giepmans and hear what he's got, currently cooking in his laboratory.
Yeah, thank you, Monica, for the kind introduction, and also thank you, Jing. So what I decided today is actually do it in 2 pieces. One focusing on what we call monotomy, which is just a routine technique in our lab. And I will also focus on electron microscopy sample prep, just to show you that it's not difficult, it's really easy. Actually, if you want to have your electron microscopy done, type 1 diabetes related, we are a core unit from the NPOT Center, so you can just send your samples, and we send a link back to show what's the electron microscopy. In the second part, I will actually come back to one of the points Jing mentioned. How can we now label our structures? And I think we find a new technique that might be really helpful in electron microscopy. So that's the part where I would say don't try it at home. Yeah, so it's still under construction, unpublished, but I think it's very promising. At least I'm very enthusiastic about it. So let's see what other people think about it. You see my opening slide also with my affiliation, and you already saw an islet of Langerhans. We zoom in, and this view, I saw some insulin granules. Probably Jing might recognize some of these things as well, but this is really difficult actually for people that are not doing electron microscopy on a daily basis. So Jing and I can read these images, might be more difficult for others. So let's go back actually only 150 years in time. This is approximately what Paul Langerhans saw when he was investigating the pancreas. So he discovered these clumps of cells. For you, again, I think it's very difficult to see the islet of Langerhans. But of course, what we are used to in light microscopy is that we have all these probes, and now mainly the use of fluorescent probes. If I would have told you that the insulin is in green, the glucagon is in red, and this would be an immunostaining with host showing the nuclei, everybody can easily make out the different cell types. Also, when Jing showed actually with the scanning electron microscope the different cell types, for me that was difficult to interpret, and I had to believe Jing on her word, which I do, Jing, definitely. But you see these different characteristics, but also when you want to look at molecules, you want to probe them. Yeah, so Langerhans. So my question actually is, can we do something like that for electron microscopy? And now, because I know not everybody has a background in electron microscopy, just a peek inside the lab with this movie and explaining the anatomy project in like 3 minutes or so.
Dit is Ben Giepmans, onderzoeker van de afdeling celbiologie, en hij doet onderzoek naar type 1 diabetes. Patiënten met type 1 diabetes moeten insuline injecteren, zodat de insuline producerende cellen in de handweefklier om onduidelijke redenen gestuurd worden. En onderzoek gebruikt hij onder andere elektronenmicroscopie, de techniek waarbij je wel tot 100.000 keer kunt inzoomen op biologisch weefsel. Om weefsels te kunnen kijken, moeten ze eerst gefixeerd worden. Dat kan met chemische processen of met bijvoorbeeld bevriezing. Daarna kan het weefsel ingebed worden in een goed materiaal. Vervolgens wordt het in hele dunne koeks gesneden, zo dun dat elektronen er doorheen kunnen. Als we 1 miljoen van deze dunne plakjes op elkaar leggen, is er ongeveer 5 centimeter. Dit is 1 van de allereerste ruimtelijke proben, gemaakt door antinucleaire. In de 20e eeuw is veel gebruik gemaakt van elektronenmicroscopie in 2011 is deze uit roulatie genomen. Heel erg goed wordt in de vacuümkamers. De elektronen die van boven komen, kunnen dus alleen worden tegengehouden door het weefsel. Is er een grote dichtheid van materiaal, kunnen we de elektronen aan de onderkant van het apparaat niet meer zien. Het beeld is zwart. Is er geen materiaal, dan worden de elektronen op het vel gedetecteerd, wat resulteert in een witbeeld. En grijs voor alles wat ertussenin zit. Maar een moderne elektronenmicroscoop is bijvoorbeeld deze. Met Giepmans veel tijd in. Deze microscoop is geheel computergestuurd. Je kunt dus een camera binnen in de microscoop kijken en daarna de beelden op het scherm verschijnen. filter, predominant in de Het probleem van de microscopie is dat de plaatjes zo gedetailleerd worden, lastig is big picture te vinden. Net als bij foto's, waar je op grote afstand je huis niet herkent, dichtbij niet meer weet naar welke strik je kijkt. Daarom hebben wij samen met het UMCG een procedure ontwikkeld die we anatomie noemen. Hiermee kun je losse beelden aan elkaar plakken, zodat je een heel groot en heel gedetailleerd beeld krijgt. Een foto van een paar miljard pixels. Deze techniek is nu nog uniek toe, maar ook in het onderwijs van medische studenten zullen we het veel voordeel toepassen om ideeën te krijgen van cellen, beestjes en echt uitzien. Heel anders dan in plaatjes in tekstboeken. Daarom hebben we de techniek hier genoemd.
So that was an overview, a movie of approximately 10 years ago, where we predicted that we would use this for education, amongst others. What you see in this slide is that we have MPOT samples shipped from Florida, the MPOT biobank to Groningen. We prepare these samples as you saw in the movie, and then we create these zoomable images and just put a link back online. So everywhere in the world, if you want to have electron microscopy from a certain donor, you just click on the donor number. First go to the website, anatomy.org, then we can go to the MPOT part, and you just click on the donor number and you have your electron microscopy instantly. Yeah, so electron microscopy for MPOT is not difficult. You just go to the website and you can go to the zoomable images and address these data sets with your particular questions. Yeah, so that's what we did previously. Everything is up and running. We are extending the database at a regular base. We still get samples from Florida, image them and add them to the database. Yeah, so from biobank to database. The nice thing is that it can be reused. Yeah, so in this case, I showed two publications where Bart Roep used the database just for illustration purposes. But Amelia Linneman actually went back and asked her questions regarding autophagy in human samples, also using that database. Yeah, so it's just available. It's kind of fair. Yeah. So you're welcome to use it, but also if you have questions related to that, we are also welcome to image and add your samples to the database. So that part, the routine part, to summarize, an anatomy, that stands for nano anatomy. It's like a Google tissue, benefits that you have an overview in a high resolution. Rare events might be imaged. Also, it's difficult, as Jing showed, also to image the cilia. There are only a few, but if you image the whole cross-section of an islet, yes, you have chances that you might see that. You have an unbiased examination. We are examining behind the computer, no longer the microscopist behind the microscope. Open access raw data sharing. I showed you examples of reuse, and it's a routine technique. But it's grayscale, like Paul Langerhans. Yeah, so everybody has to believe our expert electron microscopy interpretation. So I think that's a drawback, and therefore we took another step. This is a typical map I showed you, and we addressed, can we now actually image better, that we also, like Paul Langerhans, get colorful information in the electron microscope. So compare it with DIC in your light microscope, a fluorescent electron image, or a color EM image. Yeah, so this is what we want, what I'm now showing. Can we do that? Yeah, a little bit of spoiler, because this is really data, and I will show you how we came to that. So back to the principles of electron microscopy. Can you see my mouse, Monica? Can you?
Yes.
Yeah. So the images Jing showed earlier, she used incident electrons, and in her typical images, she was looking at secondary electrons. These are electrons that belong to the specimen, but are kicked out of the shell of the atom because of these incident electrons. These have low energy, and therefore Jing was able to capture them with a special detector. Yeah, so really electrons from the sample. However, if there's a vacancy, this is a very unstable formation of the atom, and this vacancy will be filled with an electron of a higher shell, so with higher energy. Yeah, so a higher energy electron will fill the vacancy. Higher energy, lower energy, so there's a surplus of energy, and a surplus of energy is released as a specific X-ray. So in energy dispersive X-ray imaging, if we know the energy of the X-ray, we know which atom is present. So by simply adding another X-ray detector to the microscope, we could add it to the microscope of Jing, we can detect these X-rays. If we then look at the spectrum, depending on the peak of the X-rays, we know which atoms are present. So in this case, if we have a 9.7 peak, that correlates with gold being present. Yeah, and if we have like a 23 peak, like this red one, that corresponds with cadmium present. So the moment we have all the peaks and we deconvolve them to atoms, we can actually determine where the different atoms are located. To exemplify this, this is again a beta cell. This is the nucleus labeled with some gold particles, and the insulin particles we labeled with cadmium containing quantum dots. So if we now look at the cadmium signal, yes, indeed, the insulin granules light up. If you look at the gold signal, it's indeed in the heterochromatin. Yeah, so we get color now. You don't have to believe us anymore, us electron microscopy experts. No, we now go back to the data from the microscope that tells us what we are looking at. Yeah, so I think that's a major improvement. So diabetes related, we actually saw that near the exocrine pancreas, there are some cells that have both vesicles with the exocrine signature, with the glucagon signature, and an insulin signature. So type 1 diabetes related, this has led to the start in our lab to a project where we address if the exocrine pancreas is involved in evoking endocrine beta cell stress. Yeah, so we actually have a live model for that. I will not go into that, but that's what we got out of this project. So this was actually my first slide. I introduced nanotomy, and I also introduced color EM, as we call it, so using X-ray imaging. And now our question is this so powerful? Can we combine that to get more information? So going large scale in the electron microscopy image, but also going large scale in the X-ray image. So you already see that the heterochromatin is now easily recognizable as the different cell types. These granules are different from these ones. These are red, these are purple, these are a little bit bigger. And that's only based on the nitrogen, phosphorus, and sulfur signals. And of course, we can zoom in. It's electron microscopy. This is taken at 10 nanometer pixel size. You see the endoplasmatic reticulum. The nucleolus actually also nicely pops out. This is probably an infiltrating leukocyte. So we get a bunch of information. So we have one electron image, and then we have 4,000 images with different energy levels. So if we just walk the energy spectra, looking at X-ray, we get different images. So that might help us to discriminate. So while we are scanning the spectrum, you see a change in the image depending on which X-ray are detected at that particular X-ray level. So we can use this to discriminate. So that's what we do. We now not only analyze one electron image, but analyze 4,096 2K by 2K images plus the electron image. That's way too much data. So we do some data clustering, and then we come up to end members, and these end members have unique fingerprints. And if you overlay that again over the electron image, we now see a particular fingerprint, the heterochromatin again in green, of glucagon particles, and what is it, light blue, of the exocrine particles in blue, the insulin particles, granules, and in red. So this helps us actually to automatically identify the information in our large-scale EM images. So we can overlay that with the electron microscopy, and then automatically, like Jing wants to do with the cilia, which proteins are present. And we cannot use this method for protein identification, but maybe using like the quantum dots, we get a wealth of information from the data. Yeah? So no longer only interpretation. So that's what we call identify. We can now identify using our EDX signals. I think it has a high potential for creating masks that allow to segment New fingerprints may also uncover new structures. We already see that in our other brain project. And we might not only look at endogenous signals, nitrogen, phosphorus, sulfur, but this might also be applicable for probes like the gold and the quantum dots I showed you, but also for certain stains, osmium, you name it. But then the question also comes to label or not to label. Yeah, so I'm in microscopy now for 20 years. We focused on creating new probes for light and electron microscopy, including the very nice text, of course, for live cell imaging, the M-Fruits, the M-Cherry, M-Scarlet these days. But I also know that's very difficult to make your probes. And with this kind of imaging, there's a wealth of information just from the endogenous atoms that are present as well. And so we don't need to label to get a lot of information. This information we actually then simplify, as I mentioned to you, to get the end members. And I think this will really help to analyze all these very data-heavy maps we are creating right now that are very heterogeneous. But yeah, this will be a method to be able to nail down what we can get from additional information from the NPOT EM dataset. And then back to this EM datasets, as I reviewed the nanotomy, that's ready to go, that's standard. No need to fear to use that overview in high resolution, available for everybody. So I also ask you, if you're interested, to visit the website. And with that, I think, yeah, I pitched two of the projects we have, which is, of course, all teamwork, as is listed here with all the people involved. Long-standing collaboration in new techniques with a technical university in Delft. And with that, I also would like to thank you for your attention to this talk. Thank you very much.
Thank you. And I'll just shout out that on this slide we can see there's a call for DM interested in DMDA, 1 PhD student and 2 postdocs available. So that is an exciting opportunity. I'm going to open it up to questions. I have a couple of questions myself. I mean, this is fabulous work from both of you. I mean, it's really so important to kind of, illustrate the morphology here, both in normal and T1D tissues. Are you planning to go back into the NPOD library to gain deeper information from all these samples that are pre-existing? And if so, how long might that take to kind of really, bring these all online with the new technology?
So the drawback of the new technology is that the scanning takes approximately 100 times longer. Big data sets I showed you takes 8 days to acquire one data set.
Wow.
To go back to the samples, I don't think that's the way to go. What we intend to do is using this color signals for machine learning, deep learning, overlaying that with the electron image, and then be able to see all the different organelles and stuff like that. But that will be a second step. And if we reach that, we would be able to use the grayscale images that are existing indeed.
Right.
Run on a few algorithms, and then also there add color. Yeah, but that's then predicted presence of end members. We will not scan these for the X-rays present at the moment because that is too slow. Yeah,
I see. Yeah, that was one of my questions. How can these optical data be, quantified? Do you have an algorithm yet? Is it, it looks like you are heading towards AI ML ready. And, we know the PancBase U24 grant that's just been offered by NIH, has potentially some room for making data, AI ML ready. So maybe, who knows? I know Pancreas and the HPAP, group perhaps has some room for some of this work. I wondered, and then I just wondered, has anyone done this level of work on, and this may be, just too hard, but on the TCR BCR component of some of the, immune cells present in type 1 diabetic patients?
So at this time, I think we are the only lab exploring this technique for applications in electron microscopy.
Mhm.
I don't think it has been used more widely, but that might be maybe more something, a question for Ying, because with the scanning technique Ying showed,
Mhm.
you might actually look at this immunological synapses, which you're then also looking at. If I understand well, correct, Ying? That might be feasible with the technique you showed.
Yeah, I think so. I mean, with immune infiltrates, we should be able to capture them on the surface of the islet specimen. We just haven't gotten our hands on any type 1 donor islets to see. I mean, it's hard to preserve their structural integrity, right? I mean,
Right. It's so hard.
but yes, theoretically, that is a possibility. I for one, am very excited about the elemental analysis aspect. I think that, you know, combined with what we can do, you know, labeling different organelles with different signatures of their elemental composition, I think that's incredibly powerful.
Yeah, no, I mean, it looks like you're really at the frontier of sort of opening up a whole new, you know, landscape of opportunity here, both of you. And I love the fact that you're open to, you know, offering this up to, you know, the scientific community in general. If you have some kind of aspect in your project that may benefit by this added information, you know, looks like the Giepmans lab is there to help make that happen. I guess someone's asking, will you be presenting at any of the meetings this year, ADA?
I'm not going to ADA, but the work from our lab is recently published, so that's widely available. I've already fielded some interest from other potential collaborators on this, you know, scanning EM approach, and it's
Sure.
the exciting thing is it's not just islets, you know, any kind of solid organ cilia on the surface of a cell could be scanned using our technology. It's not very high throughput, but what we could offer is a much higher resolution view of the surface organelles. So yeah, and I would say our work is not just the implication of this approach, and mine and both bins is not just limited to islets, right? It's any tissue.
Right.
So that's the exciting thing.
Yeah, it's a frontier. And you know, in terms of let's sort of circle back. Do you have any postdoc or graduate student positions available?
Yes, we're recruiting. I didn't put it on our slide, but of course, we're always happy to meet and have new members join. Anybody interested in imaging science or, you know, diabetes or islet biology? Absolutely.
Yeah. Fantastic. I guess I'll sort of like drill down on one little detail. You know, kind of highlighted the fact that there's a left-handed helix in human cilia. What about mice?
In mice as well, we see predominantly left-handed, which is interesting because in other tissues, for example, kidney cells, at least cultured kidney cell lines, what's been reported is more of a right predominant helix, and we really don't know what that means.
What is that, like an MDCK cell line?
Yeah, just based off of a couple of electron tomography structures that we have, those have a right-handed helix. You know, in terms of allowing the structure to wind and unwind and change its length, you know, the left versus right should be able to do it equally well. But you know, general biology too, in plants like Arabidopsis, there's a chirality to the way the plant, you know, twists and winds about its essential axis, and that can actually be induced to change depending on the growth condition. So that's really fascinating. But as far as the function of a left versus right helix, we at this point don't know.
Yeah, it's
But
really
potentially be reversible onto itself as well. If you can sort of imagine if it unwinds completely and all the filaments become straightened and then it twists back down, it could go the other way.
Yeah, some flexibility. And so then that kind of just leads to the fact that you're going to look in, you know, normal and disease states to kind of interpret if there are changes, correct?
Yeah, I mean, there must be a conserved purpose for it being left predominant in islet tissue. Right. Maybe with the way the cells packing it in the extracellular space. Yeah. a lot of speculation at this point.
Oh yeah, there's a lot of speculation before the paper before the work is completed. Let's see. I wonder if Dr. Hughes could elaborate on the chicken egg question of the relationship between ciliary structure and damage versus high glucose.
Yeah, that's something would be really interesting to test the causality. You know, the cilia has been, so there's been a lot of correlative structure studies out of, you know, both human and mouse islet literature saying, well, in diabetes, particularly type 2, I think that's the more studied of the two, that you have altered gene expression, for example, of cilia genes in human type 2 donors. Exactly what's the causation in there, we can't extrapolate from those kind of studies. But we also know that, you know, cilia is actually a pretty fragile structure. And the morphology could be damaged with, you know, the environmental cellular stress. And so what causes what is something very interesting, and we could be in a position to test it experimentally. For example, if you know, give the cells high glucose, chronic toxic conditions, you know, can you induce change in the cilium and therefore alter its downstream function? That would be a very interesting hypothesis to pursue for sure.
Yeah, that sounds like a fun project right there. She said, thank you. And Dr. Giepmans is announcing his next US meeting will be GRC, VEM, microscopy, and then NPOD in February 24th. So, and I guess I would say at this point, you know, if there's any other questions, just let us know right now. Otherwise, you can reach out directly and ask them offline regarding collaborations or tissue, you know, questions, microscopy questions, etc. But otherwise, this was really informative and excellent. So exciting to watch what comes next out of both your laboratories, and just looking to, I mean, as all these data become sort of quantifiable, what patterns emerge. So thank you both again for your time, and we hope to talk to you again in the future. Have a great rest of your day.
Thank you, Monica.
Thanks for having us.
All right, thank you. Bye-bye.
Bye.